So we're on the hour, so I think we can begin. I'd like to welcome everyone to this first iOS seminar of 2024. For those who are new to these seminars, they're organized by the Integration of Speciation Research Network, which is funded by the European Society for Evolutionary Biology. They're just one of our various activities aimed at bringing together a diversity of people from a diversity of different research fields, all of whom are interested in the general topic of speciation. Today our topic is sexual selection and speciation. More about that in a moment. The next seminar will be next month, March the 5th, and there we're looking at paleontological views of speciation. We have two great speakers lined up. We'd also just like to mention that the iOS network is supporting a meeting to be held at the Linnean Society in London on the 11th of April. If you're interested in attending that, either in person or online, you need to register. We'll pop a link in the chat in a moment. And we'll also pop a link in the chat to the iOS website. If you're not already on our web list, please go there and sign up so that we can keep in touch about other activities. So today we have two great speakers, very much looking forward to hearing what they have to say. The format of the meeting, as usual, will be two seminars, each of roughly 20 minutes. After each seminar, there'll be a short opportunity for direct questions about the seminar itself. And then that will take us through the first hour. At the end of the hour, we turn off the recording and we go into a kind of more general discussion. For either of those components, if you want to ask a question, please pop it into the chat and we will relay it to the speaker. So that's the background. It's a great pleasure for me to introduce Tamra Mendelson, who's the first of today's speakers. She's based at the University of Maryland, and today she's going to talk about microevolutionary approaches to speciation. Tamra's group works mainly on darters, which are very beautiful freshwater fish, as an empirical system, using them to ask questions about the evolution of signals and responses through sexual selection and their role in reproductive isolation. That's all done very much in a context of the ecology of the fish and also a context of the theory surrounding sexual selection and speciation. So the group's made really important contributions concerning, for example, the rate of reproductive isolation and the different ways in which sexual selection might contribute to the evolution of reproductive isolation. And Tamra's also written some very important reviews, or contributed to some very important reviews. And for anyone who hasn't seen the 2021 tree paper, please take a look at that on the role of sexual selection in speciation. So Tamra, over to you. It's a pleasure to have you speak to us. I'll stop my sharing.
Thank you. All right, let's see if we can get it to work. Are we good?
We're all good, thanks.
Awesome. Thank you. Thank you, Roger, for the introduction. And oh, believe me, we're going to be talking about that paper. No, no problem. And thank you, Liz and Joanna, for inviting me, and really to the whole network for hosting these talks, which is just an incredible opportunity for us with shared interests in speciation to connect. This also gave me an opportunity to get to know Tim Janicke a little better. Tim and I met once before, but we spent a couple hours in preparation discussing what we wanted to say today and how our talks might complement each other. And I really enjoyed our chats, Tim, and I hope you all enjoy both our talks. So we're both focusing on the question of whether sexual selection causes speciation. And we decided to make both our talks about empirical approaches. How do we go about addressing this question empirically, with me focusing on experimental designs at the micro end of the evolutionary continuum, and Tim talking about macroevolutionary approaches. There we go. So I'll start with this question. to sexual selection cause speciation. And simply note that if you want to know if something causes something else, if, can you see, yes, okay. If this causes that, anyway, I might not use my cursor. It seems like a good starting point is to define what you mean by this and what you mean by that, and also what you mean by cause. So I'll start with a discussion of what I mean by sexual selection, what I mean by speciation, and then I will talk about how we might be able to empirically demonstrate a causal relationship between the two. How does this cause that? And I'll finish by talking about two main approaches to testing that question at the microevolutionary scale, a forward and a reverse approach. And I want to acknowledge right off the bat that this is an almost entirely data-free talk, and any data I do show actually is not directly mine. I just was so inspired by my discussions with Tim and how we might approach this question from these different perspectives. So this is more of an idea talk, and I hope that it can generate some discussion. So first, what is this? That is, what is sexual selection? So Schuker and Farnemo, in a relatively recent paper, provided a table full of definitions that have been offered for sexual selection in the years since Darwin. I've copied them here, unformatted. And really the point, of course, is not to see them or read them, but just to understand that there are a lot of them. And we can have a lot of fun debating about, which are better than others, which are more helpful for explaining our question, or which are more accurate representations, say, of Darwin's initial vision. The point is simply that if you want to know if sexual selection causes speciation, you should decide up front what you mean by sexual selection. Then you can use that concept to guide your experimental design. So here's one that I like from the Schuker and Farnemo's paper: any selection that arises from fitness differences associated with non-random success in the competition for access to gametes for fertilization. Here's another one from Gil Rosenthal in his mate choice book in 2017. Sexual selection is a special case of natural selection, differential reproductive success due to the ability to secure matings and or fertilization. So you can note the subtle distinctions. Schuker and Farnemo specifically focus on gametes, competition for gametes. Gil is a little more broad, focusing on matings and fertilization. I'm not really going to commit, but you'll see in a few slides how I've tried to incorporate them into an experimental framework. This is another helpful addition I found from Schuker and Farnemo's paper. It's a table adapted from Anderson et al.'s 1994 book on sexual selection. And it's helpful because it reminds us that there are many mechanisms of sexual selection. So while they may all have differential competition for mates or gametes in common, they all generate differential access to mates and gametes, but there are nonetheless many different ways that sexual selection can occur and many ways that they might lead to speciation. So we can keep our eyes and ears open for example, more than just female mate choice for exaggerated male secondary sexual traits. And so speaking of traits, this table is also nice because it seems to advocate for a trait-centric view, which might help us better understand the relationship between sexual selection and speciation. Differential fitness is a result of trait differences. That's also how we identify the vast majority of species, like dung beetles that differ in their horn morphology, or jumping spiders that differ in abdominal patterns. So it's really these traits that allow us to identify the relationship between sexual selection and speciation. And you'll see that I'm also going to take a phenotype-centered view when talking about causation. All right, so we talked about this, that being sexual selection. How about that, which is speciation? So here are a few definitions of speciation that I grabbed off my office bookshelf back there, and actually one from my lecture slides. And just notice what they have in common. So Freeman et al., the biology textbook actually right there. The evolution of two or more, speciation is the evolution of two or more distinct species from a single ancestral species. From Zimmer and Emlyn, we have the evolutionary process by which new species arise. Speciation causes one evolutionary lineage to split into two or more. Oops, I forgot a parentheses there. And then from my own lecture, which you never know what you're going to get with me in lecture, but this is the one I've been using for a long time now. The process by which different species arise causes an increase in biological diversity. And so notice what these definitions have in common. In all of these cases, the buck has been passed to the much trickier question of what is a species? So the origin of what is what we're talking about. So even more than potentially definitions of sexual selection is the number of different concepts of species. So these are just a few offered by Jody Hay in 2001, which he then credits to Rick Maiden in 1997. So to ask whether sexual selection causes speciation, it seems to me like you need to pick a species concept, because are you asking whether traits that evolve because of their effect on mating success or fertilization success lead to 2 or more biological species, 2 or more ecological species, 2 or more taxonomic species? It turns out we actually have sort of an answer to the question of what species concepts biologists actually use. So these data are from a survey conducted by Stankowski and Ravenet. I have not met them yet. So if you're here, hi, look forward to meeting you someday. So they conducted a survey of 400 biologists and asked them various questions about themselves, including, do you study speciation? At what evolutionary scale do you place your research? What is your favorite what is your favorite species concept? And what really popped out is that it varies, even among speciation biologists. So yes, I study speciation. No, not directly. Here's at the microevolutionary scale. So the blue is the biological species concept. Red are genetic species or evolutionary species concept. Light blue is phylogenetic species concept. You can see a lot of variation. But what pops out to me is basically these most commonly used. The biological species concept, groups of interbreeding natural populations that are reproductively isolated from other such groups from Meyer. The evolutionary species concept, an independently evolving group of organisms genetically and phenotypically distinct from other such groups. And the phylogenetic, the smallest exclusive monophyletic group of common ancestry. It also seems that people who use the biological species concept are generally more likely to be working at a microevolutionary scale. And people who are using the evolutionary or phylogenetic species concepts are more likely to be working at a macroevolutionary scale. So that maybe is not surprising, maybe that's just a truism, but the beauty of speciation is that it really applies at all these scales. It's the link between microevolution and macroevolution. So people who study speciation are going to be using a mix of different species concepts. And so any definition of speciation that's useful for asking the question, does sexual selection cause speciation, should potentially accommodate these varying perspectives. And I tried to do that, as did my friend Becca. All right, so that was that. We did this, sexual selection. That was speciation. So let's talk about causation. So first of all, I just want to say by asking whether sexual selection causes speciation, not necessarily asking whether it's the only cause, just whether there is a causal relationship at all. And, the classical way of demonstrating cause is to conduct an experiment where you hold all the variables constant, except your putative causal factor. And if the internet is any indication, that usually involves some colorful fluids, like maybe you'd have a jar of blue fluid and then you'd have a treatment of green fluid, and then you'd see and that would be your only cause, the only varying factor, and you'd see what effect they have. But that's really hard for questions about speciation because the response variable, speciation, usually takes too long. Evolutionary biologists are comfortable with that constraint, just makes us work all that much harder to come up with compelling empirical evidence. So, with respect to causation, this is a suggestion that my good friend Becca Safran and I came up with. We wrote this review that Roger referred to, Speciation by Sexual Selection, 20 Years of Progress. We were celebrating the 20th anniversary of Panhuis et al.'s seminal contribution, which Roger was a part of in 2001. And we said that sexual selection contributes to speciation when a change in heritable phenotypes that affect mating or fertilization success within a population leads to 2 or more distinct forms separated by geographic or reproductive barriers. So we used contributes to instead of causes to emphasize that sexual selection doesn't have to be the only process driving speciation in order to have an effect. You can see our definition of sexual selection lurking in the middle there. It's a phenotype that affects mating or fertilization success. And you can see that we're trying to be pluralistic about species concepts. Some may require reproductive isolation as the defining feature of speciation. Others may be happy with genetically, phylogenetically, taxonomically distinct forms. And usually if there isn't a reproductive barrier, then those forms are geographically isolated. Okay, but for preparing for this talk, I actually noticed something and I want to tweak this just a little to be explicitly, perhaps pedantically clear. So instead, sexual selection contributes to speciation when the frequency of a heritable phenotype changes in a population due to its effect on mating or fertilization success, resulting in two or more distinct forms separated by geographic or reproductive barriers. Just want to note the two causal relationships emphasized in this new version that were implied here, but not explicit. So the phenotype in question is evolving because of sexual selection. And new species arise because of those phenotypes. So again, I think they're both implied in the original, but as I'm talking about causation here, I want it to be very explicit. And that can be summarized here with blue arrows representing causal relationships. So we can test sexual selection, however you define it, causes a change in a phenotype, and that change in phenotype causes the origin of species, however you define them, by diagnosable taxonomic forms, reproductive isolation, both or neither. So then the microevolutionary approach is to demonstrate that sexual selection caused speciation. You simply show that sexual selection caused a change in a phenotype, and that phenotype difference caused either the diagnosable difference that you're using to define species, like reciprocal monophyly, or reproductive isolation. And so now I think there are maybe two main ways of at the microevolutionary scale to generating empirical evidence for these relationships. One forward and one reverse with respect to this way of looking at the question, going from sexual selection to reproductive isolation, diagnosable forms, and the other working backwards. So the forward approach. This might entail forcing a trait to evolve due to sexual selection, testing whether species emerge. So let's say you start with a population of sexually dimorphic fish where males have colorful patterns and they vary. That would be a fun thing to study. You could manipulate sexual selection by enforcing random mating in some lines versus letting them choose or compete in others. So you have sexual selection operating in some replicant lines, not others. If you see a change in phenotype between the lines, you know that the only difference was the mating regime, and you could infer the phenotypic change was due to sexual selection. Or another idea for experiments, you could seed replicate lines with females that you know, perhaps that you've selected to differ only in preferences. And so again, if you see the emergence of distinct forms, you know the only difference was female preference. That's strong empirical evidence that sexual selection caused the evolution of these distinct forms. Even better, if you can quantify differential mating success based on the trait in each generation of this experimental evolution. And maybe that's as far as you go, depending on your species concept. But if you want to demonstrate that the divergent phenotypes cause reproductive isolation, first you could ask, do the two species interbreed? Do they want to? Are they able to? And then if you want to know whether that reproductive isolation is specifically due to the trait in question that you know has evolved due to sexual selection, you could do a swap experiment, swap out one for the other, and you should rescue that attraction. And that would be strong empirical evidence that then this trait that evolved due to sexual selection is causing reproductive isolation. So that's a forward approach, and people have been doing this kind of approach in Drosophila. So experiments by Mark Blows and Howard Rundle and Rhonda Snook and Mike Ritchie and surely others that I am neglecting to mention manipulate mating pools to both disallow and allow sexual selection. So in some lines, random mating is enforced, placing one male and one female together by the researcher's choice, not the flies. And then in other lines, flies are allowed to compete, creating at least the opportunity for sexual selection. So then if different phenotypes arise, like different cuticular hydrocarbons, and these are the only differences, then that's good empirical evidence that those characters have diverged due to sexual selection. Again, you might stop there, but if you wanted to ask if those cause reproductive isolation, you would test that if these lines are reproductively isolated. And then if you want to double check that they're isolated because of the traits in question, you do a swap experiment and see if you can rescue attraction. So that would be a forward approach. And we could contrast that with a reverse approach, which makes more sense for taxa that, say, are less amenable to living in tubes. So here we would start with two species or groups that clearly differ in a phenotype. And if you're using a biological species concept, you would want to know that those two are also reproductively isolated. Then we could ask whether reproductive isolation is caused by this trait difference. So we could do the swap experiment and see if you can rescue attraction just by changing that trait. And then you can ask whether these trait differences are or were due to sexual selection within populations. And that's really the tricky part, right? Because selection may have fixed these phenotypes a long time ago. Variation might be long gone, and you might not find any evidence of it. But A, you could try, because it might be there. And B, you could artificially inflate variation by creating hybrids or dummies or animations and introduce them to each of these populations, these parent populations, and ask whether the trait that distinguishes the species is evolving due to sexual selection. So to individuals in this population or this species, are they more attracted, or are they better competitors against or with the yellow? And is this population, is blue more successful in this population? So that would be a reverse approach where you're still asking, does sexual selection cause the trait difference, and the trait difference cause reproductive isolation, but you've just gone reverse. And I think a great example from the literature of reverse approaches comes from Carrie Shaw's lab in a genus of Hawaiian crickets called Laupala. So males in this genus sing a species typical long distance attraction song, and Carrie has shown that females are very choosy about the pulse rate of male courtship song, and that's the defining difference between species songs. So some species sing a fast song, other species sing like a medium song, and some are slow. So detailed experiments by a technician in the lab at the time. So I am first author on this paper, but it's the technician got these data, so it's not really my data. Showed that, so not only do these differences in signals cause sexual isolation, because these are long distance, and so the females are very picky, and they're not going to approach if it's a fast calling species, they're not going to approach a slow call. But even within populations, so this is Saracena sings a medium pulse rate song, Coalensis sings a fast song. Even within populations, we see variation, and we would see the opportunity for sexual selection, because females do have these preference functions where females tend to prefer a medium pulse rate call, females prefer a fast one in Coalensis. And then this is just proportion responding. So to sum, does sexual selection cause speciation? Yes, if the frequency of a heritable phenotype changes in a population due to its effect on mating or fertilization success, resulting in two or more distinct forms separated by geographic or reproductive barriers. Talked about examples of a forward approach using Drosophila and a reverse approach, again at the microevolutionary level using Laupala, but we have lots of examples of these from African cichlids and darterfish and Tungara frogs and treehoppers and barn swallows. It's a tall order for any one system to have every piece of evidence in place, but that is, the delight of our job. It keeps us busy. And I hope what I've talked about here complements nicely what you'll hear from Tim, taking a more macroevolutionary approach to the question. And just speaking of delight, these are my labs over the years. And so thank you to them and to various funding sources. So how about questions? And thank you. Did I thank you for listening?
Thank you very much, Tamra. That's a really interesting start to the day. If anyone has any questions, please pop them in the chat. We have 5 minutes or so before we need to move on to Tim's talk. perhaps I could start while we wait to see if anyone else has any questions. I mean, I think you were switching between from cause to contributes to, and I think another way of putting that might be to say, to ask the question instead, does sexual selection cause reproductive isolation or some reproductive isolation, rather than cause speciation? Do you think that would be a more helpful way of looking at things?
I tried to be clear that by cause, I just want to say that it plays a causal role, that the result of sexual selection causing divergence in phenotypes is speciation, however you define it. And so by using contribute, I just wanted to emphasize that there are lots of different causes happening at the same time. So I think that cause still works, whether or not you limit it to reproductive isolation. Is that getting at your question?
Yes, I'm just thinking of the many scenarios where sexual selection might generate some reproductive isolation, but not speciation, or it might require that there is some other reproductive isolation before, caused by ecology, for example, before the sexual selection can then complete speciation, if you put it those.
Right. So then I think it is, at least for this talk or question, the speciation network, then I think it makes more sense to focus on speciation. So because you might say that sexual selection causes reproductive isolation, but that might not be sufficient for other species concepts. And so so it might, it might lead to reproductive isolation, but that's not enough for speciation. So really, it just really depends on what your definition of each is, I think.
Yeah. We have a few questions in the chat, but we can take at least one or two of those. Alicia Lackey is asking, what do you think would help us move forward most from our understanding of how and when sexual selection causes speciation? Might it be studying rare systems or different scenarios, or sorry, I'm in trouble moving down the chat. Or thinking about mechanisms more broadly than just female choice?
To my first reaction to that, I mean, that's a great question that deserves a lot of thought. So my first reaction is the latter, is thinking about mechanisms more broadly than, say, female made choice. So thinking about all the phenotypes that affect an individual's ability to access mates and or gametes. And so how do those evolve, and how can those then contribute to species differences? I find that to be an understudied, underappreciated component, and I think would help us potentially see more examples of how sexual selection contributes to speciation.
And Jim Mallet is asking if perhaps we should think of speciation being triggered by sexual selection rather than caused by sexual selection.
That's fun. I think trigger might be a special case of cause. At least that's how I would read it. When I see the word trigger, I think start or onset. And so I think of like the chronology of all the things that are changing, and is the first thing that changes sexual phenotypes, sexually selected phenotypes, and then do those cause distinct forms of reproductive isolation? So I see trigger as a subset, is sort of like one of the first things, but maybe you read trigger differently.
Thanks for that. I think we should probably move on to Tim now.
Okay.
There's several other questions in the chat, and I think we'll try to come back to some of those topics in the more general discussion. So thanks very much, Tamra, and I'll hand over to Liz now, who's going to introduce Tim.
Let me stop.
Great. Thanks so much, Tamra. That was really interesting. And while Tim is getting his slides up, it's my pleasure to introduce Dr. Tim Janicke, who is a CNRS researcher at Montpellier and is going to talk to us today about macroevolutionary perspectives in sexual selection and speciation. Tim is an evolutionary biologist broadly focused on the causes and consequences of sex-specific selection. His research really nicely leverages both experimental approaches in quite a broad array of model systems, as well as comparative analyses. And a particular focus of Tim's work has been using meta-analyses to evaluate evidence for really sort of fundamental concepts in sexual selection theory. So things like Bateman gradients, the relationship between operational sex ratio and the strength of sexual selection, and the relationship between polyandry and reproductive success. So Tim's work, I think, offers considerable insight into the conditions under which we might expect sexual selection and the variance in mating success to contribute to diversification. So Tim, thank you so much for joining us today.
Hello and good afternoon, everybody. Thank you very much for the invitation. It's a real honor for this opportunity. Though I have to admit that speciation is not really the main focus of my research. As Liz mentioned, I'm mainly interested in sex differences in the strength of sexual selection. And for this, I am often using comparative approaches, and that's why I thought the best would be maybe to talk about the question whether sexual selection causes speciation from a macroevolutionary perspective. And I guess when we think about a macroevolutionary perspective, we would like to explore how sexual selection predicts species richness across the animal or the tree of life in general. And I'm pretty sure that many of you are familiar with the hypothesis based on which we might predict the effect of sexual selection on species richness, but I would like to outline them just briefly. As Tamra already mentioned, classically, sexual selection is expected to promote speciation by mediating reproductive isolation. And more specifically, sexual selection might facilitate assortative mating and therefore divergence. But there's also another pathway how sexual selection might affect species richness, namely by preventing extinction of species. And here, sexual selection has been argued to allow the purge of deleterious alleles across the genome, a concept that is based on the genetic capture hypothesis, and thereby sexual selection might accelerate adaptation to stressful environments and therefore prevent extinction of populations. And beside these positive effects of sexual selection, there are also hypotheses arguing that sexual selection might actually have a negative effect on species richness. And to mention just a few of them, assortative mating, if it's associated with searching costs, might lead to stabilizing selection, which might limit divergence, and also sexual selection might also favor heterozygotes and therefore also limit divergence of populations. And finally, sexual selection might also promote the extinction of populations, and here more generally, sexual selection has been argued to erode standing genetic variation and therefore the population's adaptive potential. But also sexual conflict has been also empirically shown to drive populations to go extinct. So all in all, I think it's not so straightforward to predict the effect of sexual selection on species richness on a macroevolutionary scale. And in the 90s, people started to use comparative studies to explore the relationship between sexual selection and species richness or speciation rates. And in 2011, Greifeld and colleagues thought it's time to compile all the evidence that comes from comparative studies, and they conducted a meta-analysis ending up with 64 effect sizes from 22 studies. Importantly here, most of these studies, or at least half of them, come from birds. And what they found is depicted here in this so-called funnel plot. On the y-axis is the effect size, which is just a correlation coefficient describing the relationship between a certain proxy of sexual selection and species richness or speciation rate. And on the x-axis, just a sample size or the number of contrasts. And the important bit is this dark gray horizontal bar, which indicates the global effect size that they obtained, which is positive, suggesting that sexual selection overall has a positive effect on species richness or speciation rate. But the light gray area indicates the confidence interval, which overlapped with 0 in statistical analysis that accounted for some sources of non-independence in the data. So there is a weak effect, but it's not necessarily statistically different from 0. Importantly, what they found was also that the signal that primary studies detect depended on the proxy of sexual selection that was applied. And the 3 major proxies for sexual selection in the compiled studies were sexual dichromatism, mating system, and sexual size dimorphism. And here they found that those studies that used dichromatism or mating system found a stronger support for a relationship between sexual selection and speciation than those studies using sexual size dimorphism. And of course, this raises the question now, which one is now a good proxy for comparing the strength of sexual selection across species? And I would like briefly to outline the advantages and disadvantages of the most commonly used proxies for sexual selection. So as shown before, many people use sexual dichromatism as a proxy for sexual selection, which is just the sex difference in coloration. This has been intensively studied in birds. And it's quite tempting to use this proxy because it's quite easy to quantify, even though our assessment is constrained by our own color vision, which might not necessarily match the perception of the animal that we are studying. And it's a trait-based metric. And many authors argue that if you want to quantify sexual selection, we need to measure the strength of selection on traits. But this might also be actually a shortcoming of using sexual dichromatism, because of course sexual selection might not only affect coloration, but also other traits, vocal traits, olfactory traits. And sexual dichromatism might mainly reflect, or might mainly evolve in a response to mate choice rather than in competition for mating partners, contest competition. Recently, 3 years ago, a study by Kelly and colleagues provided probably the most comprehensive comparative study on the link between sexual dichromatism and speciation rates. They compiled proxies for sexual dichromatism from almost all passerine bird species, and more than or almost 6,000 species. But what they found regarding sexual dichromatism was that there's no clear relationship between sexual dichromatism and speciation rate, as indicated by these scatter plots. So are there any other proxies? Mating system is also a very popular proxy for classifying or for contrasting the strength of sexual selection across species, which might relatively be straightforward to classify a species into monogamous species and polygamous species. And one advantage might be that it does not only capture pre-copulatory episodes of selection, but also post-copulatory episodes of selection, such as sperm competition, has been argued to be stronger in polygamous species than in monogamous species. One limitation is, of course, that even in monogamous species, there's scope for sexual selection, at least at the stage of pair formation. There's mate choice and male competition going on. And so even mating system has quite some limitations, which brings me to the third popular proxy for the strength of sexual selection, which is sexual size dimorphism, which is also a trait-based metric and is quite readily accessible because data on body size is accessible for quite many species. One limitation is that, of course, sexual size dimorphism might not necessarily only arise from sexual selection, but also natural selection. And in some species, such as waders or shorebirds, sexual selection might actually select for smaller males. In these shorebirds, for instance, males that perform certain kind of flight displays are more attractive, and smaller males have been shown to perform better in this flight displays than larger males. So sexual selection can also select actually for smaller individuals, which makes it then difficult to predict the direction in which sexual selection reflects sexual size dimorphism reflects sexual selection. Interestingly, the study that I just mentioned before by Kelly and colleagues, testing the link between sexual selection and speciation in passerine species, they also used sexual size dimorphism to test whether this reflects or predicts a speciation rate. And so in fact, what they did, they used the principal component analysis. And here's it's been shown on your x-axis, the first principal component, which was loaded primarily with sexual size dimorphism. And here they found a weak positive effect of sexual size dimorphism on speciation rate in passerines. So the question is then, what does sexual size dimorphism actually reflect in terms of sexual selection? And this was the question that we tried to address recently in a meta-analysis with my former PhD student Leonard Winkler and Rob Freckleton and Tamara Mendelson, in which we compiled data from comparative studies that tested for a relationship between sexual size dimorphism and other proxies for sexual selection. And here in this forest plot, you see all the effect sizes that we obtained. The effect sizes just represent the correlation coefficient for the relationship between sexual size dimorphism and sexual selection. And overall, we found support for a relationship between sexual size dimorphism and other proxies of sexual selection. But interestingly, what we found was that sexual size dimorphism is often negatively correlated with proxies of post-copulatory sexual selection, such as relative testis size. And the authors of the primary studies argue that this might be due to a resource allocation trade-off into investment into pre versus post-copulatory competitiveness. So also sexual size dimorphism has some limitations if you want to predict the strength of sexual selection. So of course, the question arises, what are there any alternatives? And I'm afraid I don't have a clear answer with a perfect metric how we should quantify sexual selection. As Liz already mentioned, I am often using the so-called Bateman gradient or the sexual selection gradient in comparative studies to quantify strength and the difference in the strength of sexual selection. And the Bateman gradient is just a slope of a linear regression of reproductive success on mating success. And thereby it measures nothing else than the benefit of having an additional mating partner. And thereby it quantifies the fitness net return of outcompeting rivals, and which is the essence of Darwinian or pre-copulatory sexual selection. And the longer I've been working with this metric, the more and more I also became aware of its limitation. And just to mention a few of them, as I mentioned, the Bateman gradient measures the benefit of having an additional mating partner, but it does not really quantify how difficult it is to obtain a mating partner, because it does not tell us something about the actual intensity of the competition for access to mating partners. Moreover, it's just a correlation, as all selection gradients, so it does not allow inference about causality. And as I mentioned already before, it's only focusing on pre-copulatory sexual selection. And having these limitations in mind, already some time ago, me with Niels Antis, Ines Hederer, and Marc Lejeunesse, we compiled a database of Bateman gradient spanning quite broad taxonomic range, including insects, fish, amphibians, reptiles, birds, and mammals. Overall, the database comprised analysis around 100 effect sizes, 100 abatement gradients from 70 species. And more recently, together with Luca Marie-Oliac, Mike Ritchie, and Ted Morrow, we used this database to explore whether the abatement gradient predicts species richness across the sampled families, which we had 42. And what we found is just depicted here. On the x-axis, the abatement gradient, on the y-axis, estimates of species richness for all the 42 families. And for males, we found a positive effect of abatement gradient on species richness. For females, we could not detect any pattern. And this resulted in a positive relationship between the sex difference in abatement gradient and species richness, meaning that in families where the abatement gradient is steeper on males compared to females, where there's stronger sexual selection operating on males, those families are also more diverse with respect to species diversity. What I want to highlight here is that, as you can see, there's lots of scatter, there's lots of variance unexplained. And actually, I think most of the studies I've seen, comparative studies, if they found a positive effect of their proxy of sexual selection on species richness or speciation rate, most of them explained less than 5% of the variance in species richness or speciation rate. Which brings me back to the main question of the presentation, which was, does sexual selection cause speciation? And in an excellent review by Mike Ritchie in 2007, he concluded, looking across the studies currently available, supporting evidence for a role of sexual selection is not overwhelming. And I think since this review and also since the meta-analysis that I touched upon in the introduction, there have been many studies, many more studies published, and I think it would be nice to update the meta-analysis that I mentioned before to come up with a more data-driven conclusion. But my personal impression is that there's still not overwhelming evidence on a macroevolutionary scale for a role of sexual selection for speciation, just like Mike Ritchie argued already almost 20 years ago. So to conclude, I think the comparative evidence suggests that there's, if anything, a weak effect of sexual selection on species richness or speciation rate. The big question is, of course, whether this is due to a limited role of sexual selection for speciation, or whether it just reflects our difficulties in quantifying the strength of sexual selection across species. I think what is clear is that there's a massive publication bias towards certain taxa, and with certain taxa, I primarily mean birds. For instance, I am not aware of any study testing for comparative study testing for a relationship between sexual selection and speciation in plants. And one aspect that has also been neglected, I think, is that most studies testing for an effect of sexual selection on males for speciation. But I think there's accumulating evidence that sexual selection can also operate on females, even in species with conventional sex roles. And finally, I need to mention that, of course, if whenever we take a macroevolutionary approach, we always end up drawing conclusions based on correlations, which actually does not allow to establish any causal relationship. So actually, the question whether sexual selection causes speciation cannot be fully, strictly speaking, addressed by taking a macroevolutionary approach. And with this, I hope I did not draw a too pessimistic picture on the macroevolutionary evidence for link between sexual selection and speciation. I think it's just too early to come up with a final global conclusion. And I would like to take this more as an encouragement for more comparative studies on this interesting topic. And with this, I would like to thank my co-workers, especially Luca Marie-Oliac, Mike Ritchie, and Ted Morrow, that were involved in the study on the abatement gradients. And thank you for your attention, and I'm happy to take any questions.
Great. Thank you so much, Tim. That was really interesting and a fantastic, I think, compliment to Tamra's talk. If you have questions for Tim, please put them in the chat. I can lead off with one while folks are typing. So I was curious if you have a sense of how well the strength of Bateman gradients matches other sort of trait-based proxies for sexual selection. So do you see strong, steeper Bateman gradients in taxa with greater sexual dichromatism?
We did not really explore the effect on sexual dichromatism, but in 2016, when we published this database, we also tested whether the Bateman gradients or the sex difference in the Bateman gradients predicted sexual dimorphism. And there our score included not only ornamentation, but also armaments, and also behaviors such as courtship, behaviors such as song. And there we found quite a strong relationship. So the Bateman gradient clearly predicts a sexual dimorphism. And it's also related to parental care. Yeah.
So does that make you more confident about the Bateman gradient or sexual dimorphism acting as a reasonable proxy for strength of sexual selection?
I think, it's up to a debate. I think the strength of the Bateman gradient is that it's compared to all the trait-based measures, that it captures all sorts of traits to some extent. So it's not if you study, do a comparative study on birds, it might be tempting to use then sexual dichromatism, but as I mentioned before, song might also be something that is important in some species. And maybe sexual dichromatism is not the proxy that we would choose if you want to come up with a comparative study in insects. So, but there's a huge debate on whether the Bateman gradient is now a better proxy than other ones. There is another proxy which is related to the Bateman gradient, which is termed the Jones index. I got a slide for this somewhere, or maybe not. Yes, there it is. So that's the Jones index, which is a composite measure including the Bateman gradient and the variance in mating success, which is also called the opportunity for sexual selection. And if you think about the breeder's equation, that's basically it. It's the selection gradient times the variance in mating success. And maybe that's why it has also been shown in some simulations that the Jones index is actually outperforming the Bateman gradient or other proxies of sexual selection. Problem here is if to compute the Jones index, the Bateman gradients need to be computed on relativized data, and many, at least in former times, people computed the Bateman gradient on the raw data, which does not allow to compare them across species. But nowadays it's become kind of standard. So if I would have a choice, I would probably use the Jones index to conduct comparative studies.
Great, thank you. We have a question from Goran Arnqvist. Comparative studies can only address the net effect of sexual selection, so speciation minus extinction, i.e. species richness. How can we best cope with this limitation in comparative studies?
No, that's a good question. I wish I had an answer to this one. I think that's what I wanted to cover in the introduction, that if we, especially when we take species richness as a proxy for speciation, then of course we can only explore the net effect of sexual selection on speciation, and of course that's limitation. Maybe if you focus mainly on speciation rate rather than species richness, then we might explore this, can better narrow down the effect of sexual selection. Yeah, but it's of course a tricky question.
Thanks. A question from Valentina Gomez Bahaman, getting at this idea of whether our traits are really reflecting, the traits we measure are reflecting the strength of sexual selection. So I'm curious why species in which males and females have the same size or color are thought not to have an important role of sexual selection. I would think that something like the evolution of synchronized duets, for example, is under strong sexual selection. So how well, I guess more broadly, and you touched on this a bit, Tim, as well, is how well do our trait-based measures of sexual selection actually capture the strength of selection operating in a system, and how is that potentially biasing our conclusions?
It's difficult also here to come up with a general answer. I think, as I try to illustrate regarding sexual size dimorphism, it's in the end, it differs also from taxa to taxa, where the sexual size dimorphism really reflects male-male or mate competition or competition for access to mates, or whether it's actually driven by ecological factors. I think trying to find a trait-based or using trait-based estimates for the strength of sexual selection, I think it might differ depending on what kind of taxonomic group you're studying. That's why I see lots of merit in using this kind of Bateman metrics, not only the Bateman gradient, but also the opportunity for sexual selection, or even the Jones index. Yeah.
Great, thank you. Any other questions for Tim before we move on to the general discussion? Okay, great. Well, thank you so much, Tim. So what we'll do now is move into the unrecorded discussion portion of the seminar.